Hi, I'm Linnea.
I'm Bezia.
And I'm Linnea, and this is episode number 13 of the Lemma Journal Club.
Happy New Year!
The Lemma Journal Club is the show where we read every paper to tell you if you need to read it. You probably do.
We have an algae paper for you today. We are talking about the paper Diatom Ultrastructural Diversity Across Controlled and Natural Environments by Serena Flori, Felix Mikus, Eliott Flaum, Yannick Schwab, Gautam and Flora Vincent. We also have in the mobile labs team. What is that?
This is from the EMBL mobile labs lab team. They did like a lot of the flash freezing of the diatoms that they collected in the field.
But they don't say individually who are the people in the mobile labs team. They just list them as an author.
I guess so, yeah.
I've never seen anything like that before. I wonder if I can take a course on how to pronounce names.
Yeah, I was thinking about this yesterday. I don't know why, but about the person who has to announce the person who has to announce the diploma names must take a class or be a linguistics person to pronounce everybody's names correctly. Okay.
Linnea, why did you pick this paper?
So I picked this paper as a generous term. I wanted to read a paper about diatoms because, I'm interested in getting more into diatom research. I'm particularly excited about their purinoids and that they're convergently evolved. And the fact that they make this silica, I'm excited about for bioengineering pipe dreams. But I was looking for a diatom paper, and every paper I was finding was 1 million pages long, and it's the holidays, and we couldn't read a 1 million-page paper for this podcast this week. And so you found this paper, which fulfilled our criteria of being about diatoms, having some nice videos for the podcast, and being a reasonable length.
And being a recent paper.
Paper, yeah.
The way you searched for it, presumably was through Google Scholar. You wrote diatom and then like some keywords that you were interested in. And so there's two more ways to search for papers. One, which I really like to sing the praises of, is Harzing's Publish or Perish. It's a super weird way to try to find a paper. But there's harzing.com, Harzing.com. The software is called Publish or Perish. It does something different in its search algorithm for finding papers that reliably finds papers for me that I'm not finding in any other way that are often much more relevant to what I'm looking at.
Cool.
It's black magic. It's very weird. The other way, of course, to find papers is you can ask AI, which is the worst possible way to find papers. It's a kind of interesting way. Right, Google search doesn't work anymore. Google Scholar kind of works, not as well as Harzing's Publish or Perish for reasons I don't know. If anyone from Google is listening to this, buy Harzing.com and use their algorithm instead of yours. It's so much better. But if you ask AI, it gives you papers that don't really exist, right? Even today, it'll give you papers, but they kind of exist. It's kind of getting a real paper. It just gets the name wrong and the details wrong. So then you take what it gave you and you Google Scholar search that and you find the paper that it was hallucinating about. And that's actually the way that we ended up finding this paper, was from a AI hallucination. Thought there was a paper that was something else, but this was the paper that it was hallucinating.
Yeah, and I ended up finding this paper very enjoyable. So thank you for finding it. Thanks to AI for hallucinating something similar.
God.
Yeah, I guess before we get into it, do we want to say happy holidays, happy New Year?
Yep.
Last podcast of
We should have been doing it this whole time, but this will come out December 30th. So this is the last podcast of 2025.
Great. Okay.
So, I don't think I enjoyed this paper.
Oh, really?
But I wrote to myself about halfway through reading this paper that my theory about this paper is that it was ghostwritten by diatoms. That's my theory. We get almost a page and a half in. I know it's the introduction, but it's just singing the praises of diatoms.
I loved this.
For a page and a half.
The history of
diatoms are considered one of the most successful groups of microalgae on the earth. Diatoms were among the first model organisms used to study cell biology. And it just goes on and on and on.
Well, I feel like there are people who are trying to advertise diatoms as a model system.
Yeah, they go hard on. But I feel like a lot of this, I had difficulty understanding what this paper was. A lot of this feels like it belongs in a review paper. That's fine. I'm not against having review style text in a scientific paper. But the reason why I said it felt like it's written by diatoms is presumably a lot of this is for someone like me who doesn't know the system that well. And I wasn't able to learn about the system because their review is still too hedged in their nomenclature, in their abbreviations. This paper goes heavy on abbreviations.
Yeah.
And so I still was having to Google search every other word. So I think it was written by diatoms. You know, the diatoms know themselves too well.
Yeah, those are the mobile lab participants. Are the diatoms.
Yeah, that's a great theory. You can't list just a name of thousands of diatoms who go through this. So you had to write mobile labs team, and those are the diatoms. That's a great theory.
Yeah, as somebody who's more in the field, I found this introduction a little non-standard for sure, but quite enjoyable. And I learned stuff about the history of cell biology and diatoms from the intro, which was quite nice. And I felt like very readable for somebody who's in the algae biology field.
So the paper is mostly, actually the title doesn't say cryo-EXM.
Yeah, so is this a methods paper was a question that I read the whole paper and I still don't have an answer to.
I don't have an answer either. I was trying to figure out when I would cite this paper. So the paper heavily uses cryo-expansion microscopy. So it's going to use, it's going to freeze the samples, which essentially like microfractures the silica on the outside of the diatoms. They don't really go out and say that. They just say it makes it so you can now dye the diatoms with different immunofluorescence agents. But I'm imagining.
I think that they have suggestive data. They said that right after cryo freezing, they added some protein binder and there were it bound to the silica, which they assumed were fractures.
Yeah, they didn't really explain how their technique works. They didn't invent the technique.
Right, they're just applying it to diatoms.
They're just applying it to diatoms, and they find that they think that this allows immunostaining to work in diatoms where it doesn't work usually. They don't show many kinds of immunostainings, right?
Yeah.
It would be more interesting if they stained for a lot of things and look, we've really opened the field up, right? I think here it's still a question if they've opened the field, but even if they did, let's see that this completely works. It seems reasonable that this completely works and it will work for all sorts of immunostaining. And people go off and they develop, actual antibodies that will stain the actual things. Would you, if you were then doing a immunostaining protocol to these diatoms where you have a cryo step, would you cite this paper or would you cite previous cryo expansion microscopy papers? Because this is, again, it's not really a protocols paper. They don't, in the methods, they don't really detail enough how you would do this. I don't think I would cite this paper. I think I would be asked by a reviewer to please cite this paper.
Yeah.
But it's going to have a weird lifetime, right? The lifetime of this paper, of how long it's cited for, is going to be kind of a heavy side function. If it's either going to be cited for an extremely short period of time or not at all, or it's going to be cited forever, right? I don't really know. I guess that's a good gamble to take. One thing I didn't quite understand is there's the expansion microscopy part of this.
Yeah.
I hate the term expansion microscopy because it's already confused. It's part of the confusion I'm about to state. It's already confused the issue. Expansion microscopy is not microscopy, right? There's expansion, which is a sample treatment that you did, and you can apply to all sorts of things, right? You can do spatial transcriptomics via that expansion.
Yeah.
And then there's microscopy, which is just normal microscopy. So putting together the words expansion microscopy really frustrates me because you've smushed together two completely different steps of your experiment as if it was one experimental technique, as if there was an expansion microscope you could buy.
Yeah.
Right. And so that leads me to this confusion. Is the expansion portion of the step necessary to get good immunostaining?
I think yes.
You think yes? Because they show, for instance, in figure 2A, it's figure 1A.
1A.
In figure 1A, they show pre-expansion staining. It's not immunostaining, right? It's just they usually
just NHS. Yeah, so NHS will just label any protein.
Everything.
Yeah. Any amine in the.
I mean, this is how we would label tubulin in the dosage lab, right? No?
Yes, this is how we label tubulin.
When we just have tubulin and nothing else.
I don't really want to go into the details because it's not my work to talk about, but I think that expanding the samples does improve the antibody staining.
Okay. The other thing, while we're on figure 1A, this pre-expansion, post-expansion image really bothers me because they say they use the same objective and the same microscope and the same everything for these two pictures. The pictures are the exact same size on the screen. So the blurry picture on the left might just be because they blew up. Because like the actual image that they originally took, the thing on the left is much, much, much smaller.
Yeah.
Right? They presented them at different resolutions. One of them has been like bin 2 or bin 4. One of them has been scaled up. So the two images, I mean, essentially the pre-expansion image is being displayed as an 800 pixel by 800 pixel image, but it actually is only like a 100 pixel by 100 pixel image. So you see this terrible Gaussian blurring everywhere that's not actually present in the initial data.
So I think they do try and get around this with this little inset they have in the post-expansion.
I was going to say, I don't understand the point of that inset, as it's the same as the figure 1A picture. Why do I also have the inset? That's a very good,
So I mean, like you could, like if I lean in, I'm gonna move away from the microphone. But
I see.
You could lean in and see it for yourself.
This is very close to being what I want. I just want the. Yeah, I mean, it's okay. It's okay. If they had flipped the two images, so it was post-expansion and then pre-expansion, or if they had like a zoom in. Anyways, I see the problem of why they put
I think this is
like
a constant problem in the expansion world of like how do you show the pre to post and what's the right. I think they had a whole section in their methods about the scale bars. Yeah.
And then they essentially double report this scale bar each time. I got super angry about this when I first read it. And then I thought about it for about 30 seconds. I'm like, oh, okay. Where, essentially they double report the scale bar every single time. And they say the scale bar here from the microscope is this.
Yeah.
But if you were to go backwards and not expand it, the biologically relevant scale bar here is supposed to be this.
Yeah.
That's interesting.
So it's just it's a quirk of the technique, I think.
And then they show some beautiful images. This is mostly like a beautiful images paper.
It's a good, beautiful images paper, but I wouldn't say that's mostly what I got out of it.
Well, I think we'll talk about the pyrenoids in a few moments.
Yeah.
But before we get to the pyrenoids, since we're still on figure 1, something that really confused me, bothered me, made me unclear about what the actual technique, and I went and read the methods because of this, is they have a lot of images in which you have a chain of diatoms that I imagine are connected by like maybe some fused silica or something, because they don't get separated out when you're undergoing all these processes that are quite violent towards the collection of cells, right?
Yeah.
So I was surprised that you could keep a chain together. And my solution in my mind is that these chains must be like fused together or something.
Yeah.
But then they show in figure 1I bacteria interspersed throughout the diatom chain. How did those bacteria not get shoved off during all of these washes and everything?
So I think the freezing is supposed to be not that violent. Like in an ideal world, you wouldn't be getting these fractures of the silica because they do plunge freezing.
Yeah, but they do all sorts of things in which they use like a vacuum filter to concentrate the sample,
Even in the environmental, I thought in the environmental they had this mobile lab and they just plunge froze the thing, pulled it up, and then put it in this hydrogel, and that should preserve all of and everything else happens after that.
Yeah, I had a lot of difficulty getting this from the methods.
Yeah, I didn't look that closely at the methods of this paper.
So the methods were basically say following previous cryo-EXM protocol.
Yeah.
Is the main thing. And then they say laboratory cultures and field samples were concentrated using mild vacuum filtration onto cover slips coated with polyl-lysine or cell tack. That all seems pretty violent already. After letting cells settle and attach, so already I'm like, how did the chain of diatoms survive? Residual supernatant was carefully removed and blotted off using tissue paper. So a blot with tissue paper before rapidly plunging the cover slip into liquid ethane or a mix of ethane propene using a manual plunge freezing system, which I've used on microtubule samples, and it does mess the microtubules up a little bit, but not completely. Cover slips were pasted into tubes containing frozen acetone containing 0.5% formaldehyde and 0.025% glutaraldehyde. Cover slips were moved into ethanol once at room temperature and washed with increasing dilutions of ethanol.
So it's a really violent...
And there's 5 washes.
Yeah. So these bacteria, maybe they're embedded in the silica.
Yeah, that would be pretty cool.
I'm not sure what the explanation is.
I guess talking about it with you here, I didn't realize it at the time, but I actually would like some bright field images.
Bright field, yeah.
Yeah.
I mean, the whole thing is pretty wild. Given that, although I guess in this 1J, then it seems like there's a lot of artifacts from this.
Yeah, so then my concern about the whole thing, I mean, always my concern with expansion microscopy is that we have significantly altered the sample, both by freezing and then by expanding. It's really the freezing that I'm more concerned about in this particular paper than the expanding. When we're talking about expanding, I usually get concerned about changes at the molecular scale. When we're talking about freezing, I'm very concerned about changes at this mesoscopic scale, that something that was round might become hexagonal or something as like, you try to do vitreous freezing, you almost always fail, right? It's incredibly difficult to get truly vitreous freezing. And so they talk about, a little bit later on, they talk about these triangular or pyramidal or hexagonal pyrenoids. Did you see this in figure 3?
Yes.
Do you have a different figure you want to do, or is that a good figure?
We can skip to figure 3. That's fine.
What did you think of, we're always about square droplets, right? Isn't that work so cool, right? These are triangular pyrenoids.
I found this very intriguing. I do agree that there's a big asterisk because of the freezing and because of the expansion. But just the idea that there's this diversity of pyrenoid shapes is pretty exciting. And the regularity of the, I don't know if I agree with their classification of all these shapes, but of the pyramidal ones in the A panel.
But I don't think regularity convinces me here.
That is, sure. Yeah. I'm just saying that each chloroplast has a pyrenoid of the same shape. I think we can say that.
Sure.
Right? That is exciting to me.
Okay.
And I hope that that there are indeed changes in shape, but I don't know, how would we verify that? There would have to be
you can just live image this, right?
Well, it's too small is kind of what they're saying. In this unexpanded sample, you can kind of see.
Can we not use polarization microscopy for this?
Yeah, so that's the idea.
This is incredibly, it's glass. This is the slowest sample of all the slow samples. Can we use the quantum microscope from last episode?
Well, this is the chloroplasts are inside the glass, right?
And you think they're shuffling around?
They're shuffling around,
Okay. So then can we use, a normal pulse
My question about pulscope is the glass
glass
mess up the ability to see inside, right?
Okay. And then
so you need to be able to get the NHS
for the confocal pulscope. Okay. And then if we freeze and then fix, we've already ruined. Yeah. So the confocal pulscope, we would need a genetic diatom to get a live image of it.
Yeah. And so there are a few diatoms with genetic tools, but that's less exciting to me than being able to say any diatom. Yeah. But I guess if you could, in principle, show at least this one genetic diatom, you get the same pyrenoid shape.
Yeah, that would be very convincing.
Then that's a pretty convincing, okay, this technique is not, it might be messing up a lot of things in the cell, but it does seem like pyrenoid shape might be
I think
intact.
that's unfortunate too, because again, you publish that paper and who would cite that paper, right? but that would be deeply convincing to me that this approach that they're using here is a valid approach. It's to take one genetically modified diatom and show that the pyrenoid shape is the same live as it is after this freezing and expansion.
Yeah.
None of the figures have titles.
Yeah, so there are
And in general, this is another portion of why.
No, they do.
Sorry, they have titles.
They're sentences.
Are they?
Cryo-expansion microscopy enables high resolution.
Yeah, that one is. You're right. I didn't label that one in red. But the next one is characterization and identification of various cell cycle stages in both environmental and cultured samples. Yeah. And the one after that.
It is a pretty descriptive paper, right? Which was the big drawback.
Yeah, it's very pretty pictures, description. I imagine that the throughput on this technique must be very low. Because unlike episode 2, where they had cells and they had to figure out how to time order them and all this kind of stuff, here they just time order them by hand. But here they have very, very, very few samples, and it makes me think that the throughput to this system is very slow.
Yeah, interesting. I hadn't considered how few samples they had and why that was true. They are doing a cell cycle thing, so I think they just have to accidentally, these samples are not synchronized. They have to accidentally find a cell in each portion of the cell cycle.
But that was the same problem that paper 2 that we ever did had.
Yeah.
And that was a lot of what paper 2 was about we're just going to take pictures randomly. We're going to do electron microscopy in this case, but very similar problem of a fixed sample.
Yeah, so I wonder why it's so low throughput. It seems like if you could concentrate the cells, you get them all onto one glass slide. You could do hundreds of cells.
Yeah, it's a good question. The last one was low throughput because it was electron microscopy. This is light field microscopy. Why aren't there
I mean, maybe they do get messed up very easily in this process, and so you're spending a lot of time hunting for the cells that aren't messed up.
Or do you have to, like, when doing expansion microscopy, do you have to, like, isolate a cell and expand one cell at a time or something like this?
I think as long as they're far enough apart, you're fine.
Yeah, I would have expected, I guess, this would be a lot more impressive if we were looking at hundreds to thousands of cells. Just because they talk about, like, oh, they always see the nucleus on one side of the microtubules and stuff like that. But they don't have nearly the numbers. Again, it's just descriptive. They're like, this is what we see. You know, we don't have... They don't say that they don't have that many pictures, but they don't have that many pictures, so they don't make that many conclusions.
Well, they do. So in figure 3F, they have a little table of each species and the number of cells that they
the answer to most
is one.
They have one cell. So I don't really understand what's up with that.
Yeah, I wonder what's limiting on
it could be that table 1F is really about the numbers that they have for their TEM images.
I think this is supposed to be related to E, which I think they got from their fluorescence images.
Yeah.
Their immunofluorescence. And so maybe it's the segmentation that takes a long time. I could imagine that, like, if this isn't your bread and butter, that segmenting this all out is a pain, right?
Yeah, some people are segmenting still by hand in Amaris and stuff like this. And I feel like the segmentation game is really... When I was doing my PhD, I feel like everyone was doing a kind of narrow set of segmentation methods, which either took between a day and a week, depending on how you did it. Now, there's a huge range of segmentation methods that can take between a few minutes and a week. And so depending on your training and what group you're from, segmentation has either gone from trivial to still excessively hard. Of course, it's all sample dependent, but
but this is interesting. I hadn't thought of your criticism about
it's written by Diatoms.
I had definitely not thought about that one. Okay, but the shape criticism that we have about whether this is preserved through the cryo process. These TEMs, oh no, because you still freeze.
You're still freezing. And it's the freezing step that I'm really quite concerned about. Okay. That's where I'm like, then you can get some geometry, like a sphere can turn into a hexagon pretty easily by a freezing.
Yeah.
And I guess there's also, if they're doing the segmentation by hand, then there's also a bias in the user here that segmentation by hand tends to lead you to squarer segmentations.
I can't imagine they're doing it by hand.
Dude, I think a lot of biologists are doing a lot of segmentation by hand in Amaris. I think that is a very viable strategy for biologists.
I'm just going to check.
Okay.
All 3D segmentation and visualization were made in Amira.
Similar program.
Okay. That's all that they say. I just think it must be a intensity-based thresholding segmentation.
Thermo Fisher owns Amira.
Yeah.
It's a very similar program.
Okay.
Again, it doesn't necessarily mean that they still, it doesn't rule out that they might have done this by hand.
Yeah.
Which again, if this is all the cells you're doing, if this is all the data that you have, why not do it by hand, right?
Well, these microtubules would be a pain.
Where do they do microtubules by hand?
I'm saying if they were doing this by hand, but they segment microtubules in...
Do they? In figure 3, they're not showing the microtubules anymore. They're just showing...
Okay, but in figure 2, they show segmentation and microtubules.
Okay, but they don't exactly show individual microtubules here, do they? They just do the really big blobs.
Oh, that they're microtubule bundles is what you're saying?
Well, the way that they're drawn here, they're not making much of an effort to...
I'm sorry, I'm losing the figure
bottom
numbers.
Of figure 2.
2A.
Yeah, 2A.
2A.
2A is pretty impressive.
The second panel is a beautiful picture, right?
Yeah.
Like where you can see all of these little chloroplasts and this spiral of microtubules around the nucleus.
Yeah, if
each paranoid inside the chloroplast.
If that's done by hand, that's truly a labor of love.
So I think that
I don't think that that's done by hand.
Yeah,
to be
this
honest.
is an intensity-based segmentation.
Yeah.
Is my guess.
I agree with that.
But yeah, man, I mean, these algae are just so beautiful. I've been stuck in this, or not stuck, but I've been surrounded in this clammy world, right, where we have one chloroplast per algae and one pyrenoid per chloroplast, which has been a very tractable system. But look at this single algae with 20 chloroplasts, each of which has its own pyrenoid.
But not only that, they report that, like, depending on which subspecies of diatoms you look at, I don't know if subspecies is the right word, you can really get, this one has 4 complex lobe-shaped chloroplasts per cell and 4 rectangular pyrenoids.
Yep.
This one has 1 chloroplast and 1 pyrenoid. This one has 2 elongated chloroplasts and 2 pyrenoids with a lenticular shape, right? You can very much choose your own chloroplast and pyrenoid adventure, which is pretty cool. I guess the problem with the diatoms is, Dan would talk about this all the time because Dan loves diatoms. That the problem with the diatoms is that they're very untractable, right? They're surrounded by glass. How do you image something and the thing that I want to know, that I was thinking about this, is that the shapes of the chloroplasts and pyrenoids, they say, are characteristic and unique to each diatom.
Yeah.
Cool. I wonder if the silica shape of the diatom, which is kind of acting as a lens for the incoming light, I imagine, changes the unique shape requirements that allow you to optimize your chloroplast and your pyrenoid. So the chloroplast pyrenoid setup that you have is based off of the optical properties of the diatom to begin with.
Wow, what a interesting idea.
And then can you do a little simulation of based off of this shape, this is how light is going to come. This is where light is going to get focused. So then these are your optimal chloroplast configurations.
Yeah.
And
where are the chloroplasts going to localize inside the cell?
And then based off of the chloroplast localization and the size and surface area of everything that's the solution to that, what is the optimal Rubisco solution to that?
Yeah.
And can you just do it all from geometry and the incoming light?
Yeah, so there's a I think interesting but tricky part there where a lot of what chloroplasts do is not only do they harvest light, but they protect the organism.
Yeah, the photo damage sentence really threw me. You want to talk a little bit more about photo protection?
I guess my knowledge is limited, which I say a lot.
Yeah, we already used that as the title to the previous episode. You can't use that again.
There's a big problem in photosynthetic organisms of getting photo damage, right? You have all of these absorption pigments, and if you absorb too much heat, you damage your organelle and your whole cell. So a lot of chloroplast energy goes into, we have enough light, let's radiate it off,
Yeah. So there's the question of how does that go into this simulation? I don't know.
Yeah. So that's the complicating feature.
I want to just come back to what's the point of this paper? We provide in-depth validation of cryo expansion microscopy in diatoms, which again, the cryo step, the expansion step, and the microscopy step are not super related. You could say the cryo and the expansion are more related, but the whole naming thing is confusing. And we illustrate its potential for comparative cell biology in diatoms. Yeah, they do that.
I think that's a valid summary of their paper.
And then with this study, it's written by Diatoms. With this study, we hope to generate new opportunities in diatom cell biology by enabling detailed spatial resolution of cellular structures across diverse diatom species. Here's a sentence that follows that sentence, in which, again, I'm just going to make a pitch for written by Diatoms. We invite researchers, we being the diatoms, we invite researchers to rediscover diatoms as powerful model systems at the intersection of cell biology and ecology, unlocking secrets that may guide our understanding of resilience in a changing world. In fact, unlocking secrets that may guide our understanding, our being the diatom's understanding of resilience in a changing world.
Yeah, you read this with a little bit of cynicism that I think I'm reading this with a little bit of rose-colored glasses diatoms are a great system. They are understudied. The ability to study diverse diatoms is limiting our understanding of algal biology. And
dude, I think that chickens are a tremendous system that's undervalued, and I give all sorts of hints in my paper that I'm publishing now about why chickens are great. At no point do I ever say we invite researchers to rediscover chickens as a powerful model system.
Maybe if you published in Current Biology, then you would be allowed to say that. Don't you wish you were able to say that?
No.
Seems
Again, it seems like something you put in a review paper.
Okay.
It doesn't necessarily seem like overselling. It just seems like besides the point. One thing we have while we're down here, and before I forget about it, is they do data and code availability. I've spent a lot of time over the past month exploring how can you safeguard giant data sets where they're not just held by one person. Here, I thought their solution was interesting. It's not a good solution for my particular problem, but it is a good solution for how to share information if you just imagine you're going to exist for forever. They've put all their imaging data on an S3 storage bucket, and then they give you an access key, which you can use to download the data directly from the S3 storage bucket. This is a solution that I wouldn't have imagined or known about until about a month ago when I started to play with different ways to host giant data sets. I thought that was interesting.
Cool.
Yeah, is there anything else you wanted to harp on, look at?
I mean, so one of the things that they label is the photosystem 2 proteins.
Yep.
And they talk about them always being in close proximity to the pyrenoid. I'm confused about that.
Yeah, it seems, I do have a question mark on that. PS2 fully surrounds the pyrenoids, figure 1D.
I feel that that's not actually well established in pyrenoid biology. Maybe I've missed something.
I think it's just what they happen to see. I think they a lot of times just talk about things that they happen to see, and you're supposed to be like, oh, these are low number measurements.
Yeah, it's just intriguing to me because I do think some photosystem 2 proteins pull down with the pyrenoid, but in Chlamydomonas, but it's been assumed that they're not active inside the pyrenoid. And so,
whereas if they are, it's quite like, what are they actually doing? Is there some like electron chain thing that needs to happen for the pyrenoid?
Is there like photosynthesis going on inside the pyrenoid? I think the picture right now is that it's only dark reactions. And maybe I'm just like being a fool and haven't read up enough about the biology there. But I found that an interesting observation that didn't jibe with my current understanding of the Chlamydomonas pyrenoid.
Yeah, they kind of write it in a way where they're like, yeah,
this is a thing that we saw. Yeah.
But also that like maybe it didn't necessarily jibe with what they expected to see. I had a lot of difficulty sometimes when going through their taxonomy, essentially, of what they were seeing, in that this is a very abbreviation heavy paper. They use, the particular one that annoys me is microtubule organizing center, MTOC. And they actually have like a whole paragraph about halfway through the paper. It's about halfway through the paper where they talk about, oh, the MTOC used to be called the MC by Pickett Heaps, but we're not going to use MC, we're going to use MTOC. Like they waste, I'm not going to say waste. They have a pretty long discussion about this smack dab in the middle of the paper.
Yeah.
When you never needed to use any of these acronyms. And I would like to propose a journal rule that if you use an acronym such as MTOC, Microtubule Organizing Center, that counts as 4 words for your word count. Or I guess it's 3 words because microtubule is 1 word, right? You cannot use acronyms just to reduce your word count. So that but then even later, at the very end of the paper, they're still introducing new acronyms in the second to last paragraph, where I'm like, why write out the acronym, right? They say superoxidose dismutase, SOD, and glutathione S-transferase, GST, need to be localized near sites of reactive oxygen species, ROS production. Why are you introducing, they've introduced 5 different acronyms in the second to last paragraph. You're not going to use those acronyms again. Stop introducing the acronyms. Anyways, it's an acronym heavy paper. Yeah. And that made it hard for me to follow.
I hear you.
Something.
Yeah. Yeah, the other thing I found intriguing, the other thing I found intriguing about the paper is they were suggesting that diatoms could be used as a model system for cell cycle things, which I thought was wild.
Yeah, it's written by diatoms. It's just diatoms spitballing about what could make the diatoms famous. Diatoms, if you're listening, the thing that's going to make you famous, or that has made you famous, is that you're beautiful, and that you have these tremendous degrees of beautiful morphology, right? You're like a physicist's wet dream with all these unbelievable geometric shapes that you can make by very, very small genetic changes.
Yeah.
That's what you got to go after, diatoms, if you're listening.
Should we go through our few questions?
Yeah.
What do we think is intellectually beautiful about the paper?
Yeah, so I guess they found a way to stain a sample that is not staining, that was not staining well. Sorry, I said that terribly, right? And then they've produced some pretty beautiful images. So this is a way to get people to adopt a system. If that's the goal here, that seems good.
Yeah. And then I liked that they did this I found it beautiful that they did it in field samples, so that they collected diatoms and were able to, you don't have to be using these lab strains, that you can see what's happening in the native environment. And I think that opens up a lot of possibilities for how these creatures can be studied and the biology can be studied.
If you could add one thing to this paper, what would it be? What I would add, if I was going to add one thing to this paper, is just a series of likely to work immunostaining and staining things. So I'd try phalloid and see if you can get the actin. I'd try a bunch of different dyes. I'd try a bunch of immunostaining that tends to be more robust, such as I've talked about that mitochondria is TOM20 tends to be, it just works in many, many, many species. It works in everything from clammy to
Mitotracker wouldn't work in a fixed cell.
I'm trying to think. There are mitochondria dyes that work with staining, but usually you apply the dye and then you stain. And
again, like with the freezing, I'm sure we're messing up the mitochondria.
Yeah, I mean, you'd find out.
Yeah.
But the point is, I can think of, 5 to 10 things that they could have. And I would just have quickly gone through them and been like, either everything works or, some things work, some things don't, as a warning to people. That's what I would have liked for if I could add one more thing to this paper.
Yeah. Yeah, I think that this control that we talked about of trying to see if the pyrenoid shape changes, or the shape in the cryo samples was true to the unfrozen samples, would really put this a level up.
Yeah.
And wouldn't have been that hard.
I take back the thing that I want, and I'm going with you. That's the thing that I like. That's the difference between me being willing.
I guess I don't know when this paper was published.
I think it was published this year.
This year. Okay. Yeah. So I think they could have done that.
Yeah. What I said, the first thing that I would do if I was going to start studying diatoms would be to try this protocol and see for all these different dyes what works, what are my options.
Yeah.
And same for immunostaining. Just try a bunch of cheaper antibodies and see what works. What are my options? What you said just now of compared to a genetically modified diatom where you already can see the answer live, that's the difference between me bothering to try or not, right? I'm not going to try this protocol until somebody else tries to see if this protocol is messing up shape too much.
Yeah.
Because I'm just too worried.
Yeah.
Okay, and then we're on our way out. It's a summary for Asya. Let me just check my notes. I've started to put down some podcast notes for anyone who's still listening. We have a vote for what we're doing for episode 15. If you're listening to this is probably your last week that you can vote. Probably by the time you're listening to episode 14, we're already reading the paper for episode 15. You can find that vote on YouTube. You can find it on Patreon. And I have this thing that I've been meaning to say for a while. I use Patreon for all my listening of podcasts. That's why we have a Patreon. It's because I personally, that's the platform that I use to listen to things. Patreon was created by the drummer from Pomplamoose. If you've ever seen the silly little Pomplamoose videos, he's just the guy who plays all the instruments. Pomplamoose is just him and a woman who does all the singing. They make terrific music. I wonder, because he was the drummer who then just plays all the instruments ever, if he just made the whole Patreon on his own as well, or did he hire people? Because he doesn't hire anyone to play his instruments. And then also, if you are a listener to our podcast in Bangalore or Chennai, which is also called Madras, we get a little bit of IP address who's listening to our podcast, and we get some hits in Bangalore every week. So who are you? Let us know. That's my notes for the podcast. Do you have any notes before we outro?
I have no notes.
Okay.
I didn't make any. I'm sorry. I'm a bad podcast host.
I started putting down notes to try to remember some of this stuff. So our outro consists of 2 things in which we talk directly to our very young daughter.
I thought we had changed this a little bit. That we had said that we were scrapping the second question as part of an Asya, and more we were just going to summarize the paper in 2 sentences for our listeners.
Okay.
And then answer the broadly question for Asya.
Okay, so I'm doing that right now.
Am I wrong?
Oh, sure. Okay. We definitely talked about it.
Okay.
I wasn't sure that we made any permanent changes, but I've made the permanent changes. Okay, so for listeners, and this is the non-picker does this. For listeners, specifically, scientifically, what was this paper about? And this paper was specifically, scientifically, about a way to do cryo expansion microscopy, in which you take the diatoms, you freeze and thus fix the diatoms. You expand the diatoms by putting them in a hydrogel and swelling the hydrogel. And then you put them, then you do some immunostaining. So you make certain proteins glow via some tricks that maybe one day we'll put out a little tutorial on how immunostaining works. And then they image those diatoms so you can see the inside of diatoms. And this is quite remarkable because diatoms are usually very difficult to learn anything about because they've encased themselves in glass. That's what the paper is about. It's a bit of a weird paper where it's not a methods paper, but it's also not making any big scientific claims.
Somewhere in between.
Yeah. It's like a proof of concept paper is how I would put it. And
food for thought paper.
Food for thought paper. I think proof of concept. You think food for thought?
Well, because of these beautiful images, right? Like we're gonna put up this podcast with the videos and it's just I think a food for thought of seeing the inside of a diatom.
Yeah. And then for you, Linnea, it's the picker for Asya.
Oh, yeah. Why, broadly in the world, why did we, your parents, care?
Yeah.
And yeah, I mean, we cared about diatoms because of this aesthetic reason that Bez has already touched on, which is that they create these beautiful geometric shapes. And so they are in the physicist in biology zeitgeist.
And Asya, if you're listening to this, your great-grandmother took millions of pictures of diatoms in Woods Hole, just absolutely beautiful pictures that are in some repository that is probably gone already while we're recording this, let alone, 20 years from now when you might listen to this. But it was a great passion of the whole families. We had glasses all over the house covered with beautiful drawings of diatoms. We had placemats with diatoms. I kind of don't think that any of this stuff is going to be around 10 years from now. So I just tried to paint a picture for you. Sorry, go on.
No, that's true. And so we tried, I mean, this is a little bit of a tangent, but I'll go on it. We tried recently, last summer when we were in Woods Hole, we tried to isolate some algal species from the local waters. And this is something that your great-grandmother did all the time. This was her job, was to isolate these species and characterize them.
And in fact, we got some pretty cool diatoms.
We got some pretty cool diatoms, but man, did I realize how hard this is, right? Like you've got a sample of pond water. It's got, 1,000 different species in your 1 milliliter that you're looking at under the microscope. And you've got to pick up with a micropipette the one kind of diatom that you want to isolate and get that to reproduce on its own. So you have an isolated culture. Anyways, diatoms are physically beautiful and connected to your family. And then they're intellectually beautiful or hopeful in my mind because they are, this microorganism that is able to fix CO2. And one of the big fears that I have in the world is that our planet will become uninhabitable for humans. And diatoms are one avenue to be looking into for trying to solve that terrible possibility.
Do you want to talk a little bit about why diatoms have some CO2 sequestration that you're interested in?
I'm not sure what the question is. I'm sorry.
I think you're interested, and maybe you can correct me if I'm wrong. I think you're particularly interested in diatoms for CO2 sequestration on top of, your Chlamydomonas and Volvox and stuff, because diatoms sink.
Yeah, so diatoms have a silica shell so that they do sink. It takes a long time, but in theory, they can end up on the ocean floor. And once they end up on the ocean floor, they're stable for hundreds of millions of years. So if we can, accelerate that process, we can fix CO2 on the ocean floor.
Okay. So that's our episode for the day.
Our episode, a nap time episode. So sorry if it was low energy to our listeners.
The baby is sleeping. The baby Asya is asleep right now.
But we're surrounded by snow.
Yeah. Second snow of the year. It's pretty good.
Yeah. And enjoying our holiday season.
And we'll be back in the new year. Reed asks, what is a foam? And so I don't think I'm going to pick it for the next paper because I have a very particular paper that I want to do. But as we're picking papers for the next few weeks, we should be thinking about a paper that we could cover for what is a foam.
Okay, great question. I like it.
It's a good question. All right. Thank you so much for listening.
Thank you. Bye.